Everyone, I'm Kushika Sharma from 1st Division Group number 183A, and today I'm going to present a short presentation on biochemistry of muscles. Muscle is a contractile tissue which is found in animals and the function of which is to produce motion, lift heavy objects, do physical exercises and other activities. General characteristics of muscle tissue are contractility, which is the ability to shorten forcefully, excitability, which is the capacity of muscle to respond to stimulus, extensibility, which is the ability to stretch muscle tissue beyond normal resting length and still be able to contract, elasticity, which is the ability of muscle to recoil to its original resting state post stretching. Elasticity is the reason why patients develop rigor mortis after they die because the muscles naturally want to recoil back to their original lengths. Apart from these four characteristics, the other general characteristics of muscle tissue are they are highly cellular, well vascularized, responsible for most types of movement, able to contract, and muscle tissues, muscle cells and fibers can shorten and thicken. And there are three types of muscle tissue which are skeletal, cardiac and smooth muscle tissues. As we are aware that more than 20% of the muscle mass is composed of proteins. This is largely contributed by the proteins which are named as first contractile proteins, namely actin which forms the thin filament and myosin which forms the thick filament. Second, regulatory proteins which are named as troponin and tropomyosin, and the third, structural proteins which are titin, diomycin, nebulin and dystrophin. Dystrophin has the largest coding gene. Muscular proteins structure. The protein molecules form filaments, and there are two types of filaments, namely thick filament which is formed by the myosin and the thick filament which is formed by the actin, troponin and tropomyosin. So now let's first describe the structure of actin. Actin is a major constituent of thin filaments of sarcomere and it exists in two forms: monomeric G-actin which is known as the globular actin and the polymeric F-actin which is known as the filament actin. G-actin constitutes about 25% of the muscle proteins by weight, and in presence of magnesium ions, G-actin polymerizes to form an insoluble double helical F-actin with a thickness of 6 to 7 nanometer. Troponin and tropomyosin. These two are cross-linking proteins found in association with actin. Although minor in terms of mass, they are important in terms of their function. Tropomyosin, composed of two chains, attaches to F-actin in the grooves. Troponin consists of three polypeptide chains: troponin T, which binds to tropomyosin, troponin I, that inhibits F-actin myosin interaction, and the troponin C, which is the calcium binding polypeptide. TPC is comparable to calmodulin. Now, the structure of myosins. Myosins are actually a family of proteins with about 15 members. The myosin that is predominantly present in muscle is myosin second. In terms of quantity, myosin constitutes approximately 55% of muscle protein and is found in thick filaments. Myosin is composed of 6 polypeptide chains called as hexamer. It contains one pair of heavy chains and two pairs of light chains. Properties of muscle proteins. There are four basic properties which are solubility, which is known as the protein extraction, water binding ability, emulsifying capacity, and gelation. These are the functional properties exerted by muscle proteins during meat processing. They are responsible for the texture and water holding capacity of cooked meat products and account for the product quality variations. Biological role of muscle proteins. As we know that these proteins help in the muscle contraction whenever required and help to perform the required body movement by actin-myosin complex of muscles. Muscle fibers consist of thin filaments of actin, tropomyosin and troponin which make up the L or light bands, and the filaments of myosin which make up the A or dark bands. And the sliding mechanism helps in the contraction and relaxation of muscles. Now, what are the sources of energy for muscle contraction? ATP is the constant source of energy for muscle contraction and relaxation cycle. ATP can be generated by the following ways. First, by substrate level phosphorylation of glycolysis using glucose or glycogen. The substrate level phosphorylation occurs in the cytoplasm of cells during glycolysis and in the mitochondria either during the Krebs cycle. And as we all are aware with both of these processes by the knowledge we have gained during our practical classes, we can just give a read and be familiar with them. And the net yield of ATP during this process is 2 ATPs. The next second step is by oxidative phosphorylation. It is the process by which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to O2 by a series of electron carriers. This process, which takes place in mitochondria, is the major source of ATP in aerobic organisms. The third one is by creatinine phosphate. The ATP-CP system involves an interaction between two molecules, ATP and creatine phosphate or CP, within the cell. When the ATP's outermost phosphate bond is broken off, ADP is formed along with a great deal of energy, and since the body has only about 3 ounces of ATP at any one time, however, it runs out of its supply rapidly and creatinine phosphate comes to its rescue. The fourth one is by beta oxidation. Each cycle of beta oxidation shortens the fatty acid chain by 2 carbon atoms and produces one equivalent each of acetyl coenzyme A, NADH and FADH2. The acetyl coenzyme A is metabolized by the citric acid cycle to generate ATP, while the NADH and FADH2 are used by oxidative phosphorylation to generate ATP, and dozens of ATP equivalents are generated by the beta oxidation of a single long acyl chain. Now, what is the biochemical mechanism of muscle contraction? During the relaxation phase of muscle contraction, the S1 head of the myosin hydrolyzes ATP to ADP, an inorganic phosphate, and results in the formation of high energy ADP inorganic phosphate myosin complex, as we can see in this. Now, on the contraction, the muscle gets stimulated to finally form actin myosin ADP inorganic phosphate complex, and the next step is the power stroke, which drives movement of actin filaments over myosin filaments, and this is followed by the release of ADP and inorganic phosphate and a conformation change in myosin. The actin myosin complex is low in energy state. A fresh molecule of ATP now binds to the actin myosin ATP complex. Then the actin is released as myosin ATP has low affinity for actin, and this step is crucial for relaxation, which is dependent on the binding of ATP to actin myosin complexes, which is clear in this cross-bridge cycle. Now, a fresh cycle of muscle contraction and relaxation commences with the hydrolysis of ATP and the formation of ADP inorganic phosphate myosin complex, and it has to be noted that it is ultimately the ATP that is the immediate source of energy for muscle contraction. As we can see here, the thick filaments slide over the thin filaments for the muscle to contract and relax. Thank you for your attention.